Yes, I'll keep it in check. I'll
hold. Not like last week.
Oh, come on. That was just like 3 people were here. It was fine. There was a lot of swearing coming from me right in the beginning of last week. We had some extra technical difficulties. Okay. Looks like we are on YouTube. So this is the part now where we wait for people to show up and yeah. Hawa, I didn't get to ask you in return, how's things down in DC, and are you guys opened up? You look like you're in a lab.
Yeah, I'm in the lab. So we are, at least at the NIH, right now at the NHLBI, because I don't know whether it's the same from one institute to another, we are at 50% capacity.
Okay. And, so you're not actually in DC, right? It's just kind of near
Bethesda.
In Bethesda, right? Okay, I should know that.
Yeah. So, yeah, I mean, it looked a lot, at least to me, back to normal. I am coming to work. I am going grocery shopping. My child is taken care of. So,
that's important.
Yeah. I guess since then, I feel like I'm back to normal.
Yeah, I feel like that's the big one. If you have childcare, like,
yeah.
I don't know. I don't have kids, but my heart goes out to everybody struggling with that extra complication.
Yeah.
That's really hard. I don't know. I thought I was really well suited to pandemic life, and I think in a lot of ways I am. I'm sort of a homebody, and I like just hanging out at home. But it's getting hard.
Yeah. I think the length and the fact that you don't know how long it is going to last, for me, that's the part that is nerve-wracking.
Yeah. Like my parents just emailed me and my sisters and said, like, hey guys, we don't want you coming home for the holidays this year, which I totally respect. And it was up to them, but it was like,
I just had the same conversation with my parents, and
did you?
We decided the same thing, so.
Yeah, I mean, I'd sort of thought, okay, like I can quarantine for 2 weeks ahead of time, and I can, Harvard's doing tons of tests. Like, we, in academia anyway, we have access to tests, I think, probably most of us, as opposed to, it's a little harder for the general public. And, I was like, I can wear gloves on the drive down to New Jersey. Like, I know sterile technique, like it'll be fine. But I think it was just, for them, the risk is too great.
Yeah.
So I don't know. I think the holidays are normally my favorite time of year, and the ones coming up are going to be a little darker, perhaps.
And that's for Thanksgiving or also a New Year?
Yeah, sort of like the, Christmas, Hanukkah, New Year time.
Okay.
Yeah.
Okay.
I have a bunch of sisters, so we usually get together at my parents' house and just like watch the Great British Bake Off and eat a lot of food, and it's a lot of fun. All right, so we'll just wait another couple minutes while people start joining, and then we'll get started. So Becky actually made us a playlist for this sort of like waiting period for, it's, I think, songs about movement. Unfortunately, I don't have Spotify. I'm like the only millennial without Spotify, so.
So Adam was also co-creator of the playlist.
Oh, okay.
Yeah, I can't take full responsibility. I won't say credit.
We've not had to use it yet.
I'm trying to figure out if there's a way for us to embed it on the website, because I think that'd be quite funny to have like resources, and just one of the resources is a playlist. But I can only get it to be like follow someone on Spotify, and I didn't want to be like, hi everyone, follow my Spotify account, because some of my playlists are quite embarrassing. Like Adam's found them before, and it's like sad Becky, extra sad Becky. Like, Becky, you need to go running. Like,
Right. So maybe just we have to come up with a more anonymized way to share that playlist.
That's what I'm trying to figure out. Yeah.
Okay. I don't know if you're old enough to remember MySpace, but
yeah, no, I had MySpace.
Did you? Okay. Okay. That makes me feel slightly less old. But yeah, that was a good way to share your music.
True.
All right. Adam, what do you think? Shall we go? All right. I'm going to go ahead and start with the intro. People always keep coming in for the first like 5 minutes or so. Okay. Hi, everybody. Good morning or afternoon or evening. Welcome to our 23rd installment of cell migration seminars. Just a quick reminder before we get started that next week we're going to be off. As I think everybody knows, it's the US election. Hope everybody votes. So then we will see you in 2 weeks and we'll see what the world looks like then. All right, so first up we have Hawa, Racine. You tell me how to say it again.
CH. Think about CH.
Thiam?
Yes, Thiam.
Thiam, thank you. Hawa Racine Thiam, and she's a postdoctoral research fellow in the laboratory of Dr. Claire Waterman at the NIH, and she got her PhD in biophysics from the Institut Curie in Paris in the lab of Dr. Mathieu Piel. She was looking at cell migration under confinement. And Hawa's current research interests center around how immune cells integrate physical forces into molecular pathways to achieve their host defense functions. Today, she is going to be telling us about her postdoctoral work on the cellular mechanisms of netosis. Did I say that right?
Yes.
Netosis, which is an unusual type of immune cell mediated pathogen killing. And I'm really excited to learn about it. Thank you for being so gracious for my ignorance, and go ahead and share your screen when you're ready.
So don't worry at all. I am, yeah. Yeah, okay, so I'm on. Yeah, thank you very much for being here, for attending this seminar, and thanks a lot to Jennifer Adams and Becky for putting together this amazing seminar series. So whoever knows me in the audience knows that I like things that explodes or rupture. And today I will take you from the subject of cell migration to the subject of cell explosion during netosis. So this project is a result of a collaboration between our lab at the NIH, the lab of Denisa Wagner at Boston Children's Hospital, and the lab of Bob Goldman at Northwestern University. And a big part of this work has been done at the Marine Biological Laboratory in Woods Hole. And one person I would like to highlight here is Christine, who has been really my partner throughout this process. I mean, scientific partner, I should say. Yeah, throughout this process. So neutrophils are innate immune cells that are produced in the bone marrow, where they differentiate from the myeloid progenitor cells. And once they differentiate in the bone marrow, they are released into the circulation, from where they can be recruited to sites of infection by activated endothelial cells or macrophage. And at the site of infection, neutrophil, they can deploy a plethora of mechanisms to fight pathogen. And these include the phagocytosis, so the internalization and internal killing of pathogen, or degranulation, which is the release of their granules that contain cytotoxic protein to the extracellular environment, which then kills those pathogens, or the release of their decondensed DNA during netosis. So neutrophil extracellular traps, or nets, are DNA decorated with histone and antimicrobial protein, such as neutrophil elastase, or which is a serine protease. And they are released not only by neutrophil, but by other granulocytes and also mast cells or macrophage. And one particularity of those nets is that they can physically trap cells. They can physically trap pathogen, as we can see here on this electron micrograph showing this yeast cell that has the DNA of nets tightly wrapped around it. And not only they can trap and limit the mobility of those pathogens, they can also kill or neutralize them. And this has been shown for bacteria, fungi, or viruses. And consistent with that, nets, they can limit the growth of bacteria in vivo. Strikingly enough, at least for me, is the fact that the antimicrobial capacity of nets requires the DNA scaffold and histone. So all this would position netosis as an important part of innate immunity. However, nets can also be detrimental to the host. For example, in the specific case of sepsis, the high concentration of bacteria leads to the activation of platelets, which then activate neutrophil, which release a high amount of neutrophil extracellular traps. And because of the content of cytotoxic protein in those nets, this drives endothelial cell death and then tissue damage. And more evidence point towards a detrimental effect of netosis in inflammation-related disease, such as diabetes, cancer, or COVID-19. So because of that, we think it's very important to understand the mechanism of netosis. And the current model in the field is that neutrophil activation, either by specific pathogen-derived protein, or other types of what could be called, what I think the field would call danger signal, would drive neutrophil activation and the production of reactive oxygen species, which lead to the translocation of some proteins to the nucleus to drive chromatin decondensation. And two of those proteins have been proposed to be required for netosis, and one of them is neutrophil elastase, which is a serine protease, which is proposed to mediate chromatin decondensation by degrading histone. And the second protein is peptidyl arginine deaminase 4, which is proposed to mediate chromatin decondensation by citrullinating histone. And this is basically, so this is a post-translational modification which consists of a removal of positive charge. And also, so of note, the production of autoantibodies against citrullinated protein is a hallmark of several autoimmune disease. And so the citrulination of those histone is proposed to drive chromatin decondensation by decreasing the electrostatic interaction between histone and chromatin and the DNA. So once the chromatin is decondensed, it would be released from the nucleus, then extracellularly. However, this picture is mainly based on fixed time point assay and electron micrograph experiments. So whether, how, or when would PAD4 or neutrophil elastase translocate to the nucleus is unclear. The rupture or the release of DNA from the nucleus or from the cell was never visualized live. And also, if we take the DNA as a reference point, so the DNA is located inside the nucleus, and it's physically separated from the rest of the cell by the nuclear envelope, which consists of the nuclear lamina and the double lipid bilayer of the nuclear envelope. And if the DNA would breach this nuclear envelope, it would have to deal with the dense interconnected network of the cytoskeleton and the organelle before breaching the plasma membrane. So how do we go from this configuration to this one? What are the cellular and biophysical mechanisms that drive netosis? So the two specific questions that we asked here were, what are the cellular events that mediate netosis? Just because it was not known, we did not know how the cell goes from being inactivated and to releasing its DNA extracellularly. And we wanted to know the role of PAD4 in those cellular events, because again, this protein has been involved in netosis, especially for mouse neutrophil. However, it's unclear whether it's required for human neutrophil netosis, and it was also not clear which specific aspect of netosis it was required for. And the approach we took is a microscopist and cell biologist approach. We use high-resolution confocal microscopy to look at cell and nuclear morphodynamic. And we use neutrophil from different sources. We looked at mouse and human blood-derived neutrophil, as well as HL60-derived neutrophil, just in order to be able to manipulate these cells more easily. So in this movie, what we have is the HL6 derived neutrophil undergoing netosis upon ionomycin stimulation. And in red, we have the DNA. And now on this plane is the bottom plane, so the interface between the cell and the cover slip, which allows us to see the cell dynamic. And plane 3 micron above allows us to see the nucleus and the change in the nucleus. So the first event that we could observe here is that cells, when they start the process of netosis, they start by shedding those vesicles from the plasma membrane. And after shedding those vesicles, we start seeing change in the nucleus, change in the compaction states of the DNA, change in the shape of the nucleus, release of the DNA in the cytosol before the release of the DNA extracellularly. And we can quantify this in multiple cells, and we can see that we can already see that like these events, they occur in all cells that are undergoing netosis. I mean, what I mean is that like a cell that releases the DNA extracellularly would always start the process by shedding vesicles, changing the chromatin compaction, changing the shape of the nucleus, and releasing the DNA extracellularly. So since this was done in cells that are stimulated with ionomycin, and we wanted to have something that is more physiologically relevant, we exposed a primary human neutrophil to the yeast Candida albicans, which is in cyan. And what we could see was that the cell start the process by first internalizing those yeast, as we can see it here. So the cell becomes cyan itself. And then after that, we see changes in the cell itself. So we see formation of those vesicles from the plasma membrane. And then the other similar change, so the compaction of the chromatin, release of the chromatin in the cytosol, and then release of the chromatin extracellularly in the end, where then we can see it in green because it can bind this cytotoxic green DNA dye. And again, by following single cells over time, we can see that this is really a sequence of events, that the process, each cell that released the DNA extracellularly started the process by shedding vesicles, changing the compaction states of the chromatin, changing the shape of the nucleus, and then releasing the DNA extracellularly before releasing the DNA in the cytosol before releasing it extracellularly. And by quantifying, by following thousands of single cells over time with 3 different types of neutrophil and 3 different types of stimulant, we showed this well-conserved sequence of events. And because one of the first events that we observed was this formation of those vesicles, and we know that microvesicles, especially from neutrophil, they can be bioactive, they can mediate cell-cell communication, and they can even change the inflammation state of the system. So we wanted to know whether these were microvesicles or whether this was degranulation. So this process where neutrophil would just release their granules to the extracellular environment. So one thing that I show here that we did to understand this process a little bit better was to express a soluble emerald in these cells and to stimulate them to undergo netosis. And we could see that this emerald, the cytosolic protein, would go in those vesicles, also indicating that those vesicles would just contain cytosolic protein. And we have also other data showing that they contain protein that are normally in granules, so they contain just whatever was there at their formation. And we know that, we show that at the membrane of those vesicles, we have a protein that are known to be at the plasma membrane. So they are they emanate from the plasma membrane. And lastly, to show that those vesicles are physically separated from the cell, what we did was to bleach the emerald signal in those vesicles and look at the recovery over time. So what we can see is that while in the cell body we have a recovery within a second, in the microvesicle we don't see a recovery of the bleach emerald. So this indicates that those vesicles are physically separated from the cell body. So now that we know a little bit better how those vesicles are formed, and I mean what are in those vesicles and when they form, and that they are shed from those neutrophils, we wanted to come back to the DNA again as a reference point and try to understand how would the DNA escape the nucleus. And like the question was, okay, whether we see rupture of the nuclear envelope, whether those the DNA would be released from the nucleus via vesicle shedding as well, or what. So here, just to show you a piece of data showing the lamina, not the lamina, the inner nuclear envelope network that is marked by lab 2B, and the lamina network. So here we have a marker of lamin B1. So what we can see, and in blue we have the DNA. So what we can see is that the DNA would be released from the nucleus via local rupture of the nuclear envelope. So we see first a rupture of the inner nuclear envelope and the rupture of the laminar network from where the DNA spills out of the nucleus. And when we quantified those events, we saw that like the first event that happens around the nucleus, I would say, is the formation of vesicles from the ER. So we see the ER vesiculation, and this is followed by the rupture of the laminar network that we can visualize either with lamin B1 or lamin A. And then this is followed by a rupture of the inner nuclear envelope, which coincides with the rupture of the outer nuclear envelope. So this shows that the DNA is released from the nucleus via this specific, again, sequence of events that involves vesiculation of the ER, disassembly, rupture of the lamina, the inner and the outer nuclear envelope. So now the question is, once the DNA is out in the cell, how does it deal with the dense interconnected network of the cytoskeleton? So what we did was then again to just look at the cytoskeleton, and what we saw was that the actin is initially localized in the lamellipod and the uropod, but over time we lose this staining, and the marker becomes homogeneous inside the cell, showing that the actin cytoskeleton is disassembling. And then when we looked at the microtubule network, we saw the same, that initially we can see these nice bundles of microtubule in the cell, but then over time, these bundles would disappear. And then we can also, when we looked at the vimentin intermediate filament, we can see that initially, again, this forms a network in the cell. But then when netosis is stimulated, we lose this network and the signal becomes soluble. So by quantifying this again, we can see that the disassembly of the actin cytoskeleton was the first event that we could observe. And this is followed by the disassembly of vimentin intermediate filament network and the disassembly of the microtubule network. So this tells us that, well, when the DNA is released from the cytosol, there is no from the nucleus to the cytosol, there is no cytoskeleton that it has to deal with because the cytoskeleton is already disassembled. Then we wanted to know, how does it bridge the plasma membrane? Again, do we have pores that are formed? If there are pores, what would be the size of those pores? Do we have vesicles that are formed that allow the DNA to be released extracellularly? What was happening here? So to have an idea of what type of pores would form at the plasma membrane and when would they form, what we did was to add these membrane impermeable dye of different size outside of the in the media, reasoning that if a pore would form at the plasma membrane, those dyes would get in, and they would get in depending on the size of the pore. So here we are looking at a primary human neutrophil that is undergoing netosis. And then in the media, we have a calcine, which is a 0.6 kilodalton dye, and the dextran, which is a 10 kilodalton dye. And what we can see was that there is first the entry of calcine before the entry of dextran. So we see this sequential increase in plasma membrane permeability. However, this increase in plasma membrane permeability doesn't correspond with DNA being released extracellularly. So when we looked at the plasma membrane itself, in these HL60s and the DNA with histone marker, we can see that the DNA escapes the nucleus when there is a local rupture, so the formation of this huge hole at the plasma membrane. So again, by following thousands of single cells over time, 3 different types of neutrophil with 3 different types of stimulants, so ionomycin, LPS, or the yeast Candida albicans, we identified these 13 cellular events that occur when cells undergo netosis. And more importantly, they occur in this very specific temporal order. So the cells start the process by disassembling the actin cytoskeleton before forming those vesicles and disassembling the rest of the cytoskeleton and forming vesicles from the ER. And this is followed by changes in the chromatin, namely decompaction of the chromatin, change in nuclear shape, and release of the DNA in the cytosol via rupture of the nuclear envelope, which was an irreversible process here. And this allows the DNA to expand in the cytosol before the DNA being released extracellularly via rupture of the plasma membrane. So since, as I said earlier, PAD4 was involved in netosis, but it was unclear which specific cellular event it was required for, we wanted to know which of those cellular events require PAD4. So the first step that we took was to just look at PAD4 and its localization. And we show that it localizes with the DNA, so inside the nucleus, and this is a primary human neutrophil, so it has this beautiful nuclear shape, in my opinion. So when we use a neutrophil from PAD4 knockout mice, and we show that all the upstream cellular events are independent of PAD4, namely microvesicle shedding, cytoskeletal disassembly, vesiculation of the ER, change in nuclear shape, or permeabilization of the plasma membrane. However, when we look at the DNA and the nucleus, we saw that when PAD4 is absent, there is this decompaction, there is a defect in chromatin decompaction, a defect in release of the DNA in the cytosol, so defect in rupture of the nuclear envelope, and then defect in release of the DNA extracellularly. So since this was done in primary mouse neutrophil, and we wanted to know whether PAD4 was also required in human neutrophil, we generated a PAD4 CRISPR line in HL60s, and we found that there is a defect in chromatin decompaction, release of the DNA in the cytosol, and release of the DNA extracellularly. And we were able to rescue that by adding back PAD4, the wild type version. But then we were not able to rescue that if we add back the enzymatically dead, so the one that cannot citrullinate, or the NLS mutant, so the one that has a defect in localizing inside the nucleus. So, and just to show you that this is quantified, because we do quantitative cell biology, yeah. So here what we did was to identify, not only to identify those certain cellular events that occur during netosis, but to show that the protein PAD4 is important for chromatin decondensation, release of the DNA in the cytosol, and release of the DNA extracellularly. And we also show that the disassembly of the actin cytoskeleton is important for the rupture of the plasma membrane. So, and just to say, like, since we started working on this, we have been contacted by several clinicians that are interested into understanding the cellular mechanism of netosis in their specific disease state, human disease. Yeah. So just to end with, like, what are the questions that I would like to ask moving forward? And this is a little bit coming back to my, biophysical root. So the first question is, like, how are the forces that are necessary for netosis generated? And this is just because, like, the cytoskeleton is gone, and the cytoskeleton is the main force generator, at least the best known force generator in cells. So then how would you rupture the nuclear envelope or the plasma membrane without it? And then what is the mechanism and function of microvesicles? And in the long term, how do the physical properties of in vivo environment influence neutrophil decision-making to undergo netosis? So with this, I would like to end by thanking the Waterman Lab, and especially Claire, my mentor, the Wagner Lab, both Denisa and Christine, and also the Goldman Lab. Thank you very much for your attention.
Thanks, Hawa. That was really, really interesting. I had heard of netosis, but I didn't know anything about it. And it's really wild. Really, really cool. So we'll jump right into questions. We'll get through as many as we can. We're running a tiny bit short on time.
Okay.
So from Will Gibbs, we have at roughly what time point is the microtubule network degraded, and do you see netosis with PMA?
Whether I see netosis with PMA, yes, and it has been reported. The first report of netosis was done with PMA, actually. And when roughly, it's about, I would say, 2 minutes after microvesical shedding. We can go back to that. And I don't think we see, so here, I mean, the mean is at 0.9, but it has this huge spread. So it's about 1 minute around microvesical shedding. And I would not say that it's a degradation. I see disassembly. So I think to talk about degradation, I would need to see, to do a staining of microtubule and see that, there is degradation of tubulin. So I can speak of disassembly. I cannot speak yet of degradation.
Okay, great. I'm gonna ask one more question, and then maybe we can return to these at the end because I want to give Karen enough time. So from Anna Huntlacher, do you see different stages of evolution of cytoskeletal changes depending on the trigger of netosis? For example, candida versus the ionomycin, et cetera.
So we looked at the disassembly of the network with LPS and ionomycin. And as far as we could tell, we could not see a difference. So between LPS and maybe I can talk to you, Anna, later. I don't know really what you mean by evolution of the cytoskeleton. Do you mean just a disassembly or something else?
Yeah, I'm just wondering if it goes through the same pattern of changes, or are there distinct triggers that would have a different evolution? I just also want to say, really nice talk.
Thank you very much. So yeah, as far as disassembly goes, we see the same pattern of disassembly. And by disassembly, what we mean is that we have a network, like for example, here we see the actin at the uropod and at the front, and we have this inhomogeneous distribution of the actin probe, and we lose that. And we see that, if we, for example, look at G-actin, or if we look at different types of probes. So that's how we quantify the disassembly. Now, the fine characterization of the disassembly of those cytoskeletal network is definitely needed. And that would be great to see in the future.
Thank you.
All right, thank you so much. This was really interesting. I'm gonna have Karen go ahead and start sharing her screen. Hawa, if you wanna answer questions in the chat afterwards, or folks can stay and we can discuss this at the end. Okay, awesome. Okay, so now we have a second great talk. We have Karen Kasza. She is a professor of mechanical engineering at Columbia University. She received her bachelor's in physics and mathematics from University of Chicago, PhD in applied physics from Harvard University, and then did her postdoc at Sloan Kettering. So the research in Karen's lab combines approaches from biology, engineering, and physics to explore how cells self-organize into functional tissues. And so a major focus of her work is to uncover fundamental physical and biological mechanisms underlying tissue morphogenesis during embryonic development, and to leverage that understanding to build functional tissues in the lab. Thank you so much. Take it away, Karen.
Great, thanks so much. I'm really excited to be here today. The seminars the last few months have been fantastic. So today I'll tell you a little bit about some of the work from my lab at Columbia, studying kind of the biophysical mechanisms at play during tissue flows during embryonic development and morphogenesis. And so my lab, we're really interested in understanding how the really remarkable mechanical behaviors and properties of cells and tissues help really link the molecular control of cell behavior and cell fate to the actual physical and mechanical shaping of tissues and organs to really make a functional structure at the end of the day during development. I come from a soft matter physics background, and so I really think about these tissues as just really remarkable, soft, squishy materials. And in my past, spent time thinking about materials maybe epitomized by things like silly putty or foams like shaving cream, which have both solid and fluid properties and might flow depending on kind of the level of force or stress applied. And in all these cases, I was really interested in connecting the microscopic structure and dynamics of the material to the macroscopic behaviors and flows. And so, more recently, I've been focusing my attention on morphogenesis of epithelial tissues. And again, these materials are kind of extra remarkable soft squishy materials because there's active forces inside and kind of genetic programs that can control the properties of these tissues in order to allow them to change shape actively during development to build tissues and organs. And again, my lab is interested in kind of looking at the microscopic scale, looking at the proteins and cell level structure and dynamics to explain the macroscopic tissue and organ and animal level behaviors. So in my lab, we were mostly using the Drosophila fruit fly embryo as our model for studying epithelial tissue morphogenesis. If you haven't seen them, hopefully this movie is playing smoothly enough to kind of keep track of what's going on. But this is the fly embryo a few hours after it's been laid by the mother. Future head is over here on the left. Tail is over here on the right. Each of these small polygons is the outline of an individual epithelial cell. And essentially, kind of over the course of just a few hours, this epithelial tissue sheet bends and folds, stretches, flows, and remodels to lay out the basic body plan and shape of the animal. And so I just think this is, a remarkable process that, we'd like to understand, especially kind of how we kind of at a molecular level control the mechanical forces and mechanical properties of these tissues to make these tissue shape changes play out in the proper fashion. Today, I think we'll focus mostly on tissue shape changes during development and morphogenesis within the plane of the tissue sheet. So not bending and folding out of plane, but tissue shape changes in the plane where you change the dimensions of a tissue. And if you think about kind of how you might do this, if we restrict ourselves to cellular mechanisms that preserve the number of cells in the tissue, we're largely left with two dominant mechanisms at play that could give you these macroscopic changes in tissue shape. We have cell rearrangement, where cells essentially intercalate between each other in order to swap neighbors, change their local positions, leading to, if it's done in an oriented, organized way, a macroscopic change in tissue dimension. In contrast, you can also imagine that you could achieve the same kind of macroscopic change in tissue shape just by changing the shapes of all the individual cells along some direction without necessarily changing your local neighbor relationships. I really like to think about these cell rearrangements as kind of a fluid or plastic like deformation where cells change their local positions and then with that potentially lose information or their memory of where they started out and who their original neighbors were. Whereas this cell stretching type of mechanism maybe is more of an elastic solid like behavior where cells may still retain some memory about kind of their original shape and their original position and potentially go back to that later on. Okay, so if you look in the developing fruit fly embryo, you can see examples of both of these at play kind of over and over again during development. And so if we first look at about 3 hours into development, the lateral side of the embryo is the germ band epithelium. And this tissue narrows and elongates in order to kind of elongate the head to tail axis of the embryo. And during this process, the tissue remodels and flows largely through these cell rearrangements where cells locally swap neighbors within the tissue in an oriented way. Other cell stretching and cell division also play a role later in this process, but cell rearrangements play a really dominant role. In contrast, if we look at a neighboring group of cells in the amnioserosa, so another epithelium just next door to this germ band, in contrast, this tissue is going through a comparable change in tissue dimensions, but it's occurring through these really dramatic changes in cell shape, where you can see this stretching really nicely illustrated in this paper from Shane Hudson's lab. And so, I'm really interested in why these two groups of cells in these neighboring parts of the embryo behave so distinctly different, and how that's regulated. And I think this raises the question, are tissue properties within the embryo regulated to either allow or prevent cell rearrangement, tissue remodeling, and tissue flows at different regions of the embryo and at different times, in order to help accommodate these really dramatic changes in shape? Okay. So today we'll just focus on this germ band epithelium that's going through this narrowing and elongation process, where cell rearrangements play a big role. So this is the side view of the fly embryo a few hours after it's been laid. And this region I'm showing right here, highlighted by this yellow line, is going to go on to form this segmented part of the adult fly body. And over the course of a couple hours, it narrows along this vertical axis and elongates along this head to tail axis, and then folds over on itself because the whole tissue is encased in an eggshell. So it elongates and folds over. This is a really rapid process. We get a twofold elongation in tissue length in about 2 hours, where most of that actually happens in the first 30 minutes of the process. If you take a closer look at some of the cell rearrangements occurring during this event, they look something like this. So this epithelium, the rule is that if you want to swap your position with your neighbor, you're not allowed to open up a hole in the tissue sheet. So you go through these what are called T1 or rosette events, where you, for example, shrink a contact between two neighboring cells, and then bring those cells together at a higher order vertex, and then you form a new contact, allowing the cells to swap neighbors and produce a local unit of tissue elongation in this process. And from a lot of really beautiful work from a number of labs over the last few decades, we know that myosin 2 and actomyosin contractility play key roles in driving and orienting these cell rearrangements, so they produce elongation along the head to tail axis of the embryo. And you can see here in this image that myosin 2 is enriched on these interfaces, which are vertical in this image, which are oriented between edges between anterior and posterior cell neighbors, and it's contractility at these edges, which we think is driving these local rearrangements in the tissue. So internal forces generated by cells driving these rearrangements and tissue flows. Okay. So we've spent a lot of time thinking about this process already, but we wanted to take a closer look at these cell rearrangements and considering maybe how the cell rearrangements and tissue fluidity are changing at different points in the process. So what we do is we take movies, we track the cells, and we keep track of every time a cell swaps neighbors and count that as a cell rearrangement. And if we do that, we can then plot the instantaneous rate of cell rearrangements in the tissue as a function of time. And what we see is this: we get a rapid burst of cell rearrangements in the tissue that corresponds with the macroscopic elongation of the tissue along the head to tail axis, which I'm plotting here. And so if you take a little bit of a closer look at this, you might want to break this up into 3 different periods. One is early on, well before cell rearrangements and tissue flow and tissue elongation begins at t = 0. We have very low levels of cell rearrangement in the tissue. Then we transition to this phase where we have this rapid burst of cell rearrangements, and then we slow back down again to having very low levels of cell rearrangement again. And so kind of at a mechanical level, you could imagine a lot of different explanations for this particular behavior. But one that we got interested in was whether the tissue might be becoming more fluid-like exactly during this stage of development, exactly in the germ band and maybe not in the neighboring amnios serosa, to accommodate this rapid tissue remodeling and tissue flow. And the time we were thinking about this, you know, there were kind of difficulties in actually going in and directly measuring the mechanical properties of epithelial tissues inside this tiny, soft embryo encased in an eggshell. So we decided to gain some insight from simple physical models of epithelial tissues, the Vertex models, which have been used really quite successfully over the past decades to describe the stable packings of cells in epithelia and dynamic rearrangements in epithelial tissues. And so, you know, we're going to use a kind of very simple version of these Vertex models where we take our epithelium, focus on this apical side where much of the actin and myosin and adhesion, cell adhesions are in the fly embryo, and think about this as a collection of cells where each cell is considered as a polygon. That's a collection of edges and vertices. And then the energy of each cell then has contributions that depend on the shape of the cell properties, and the interactions between cells in the tissue. And so typically these types of Vertex models have one term that describes a preferred area of cells in the tissue, so some type of area elasticity, and then some other terms that describe some type of preferred kind of contacts between the cells in the tissue that kind of give us a preferred perimeter, you might say. And so between the preferred area and preferred perimeter or length of contacts, that defines kind of a preferred shape of cells that minimizes the energy. Okay, and from a lot of work from a number of different cell types and systems, you know, we typically think about these terms over here as, you know, a line tension, which depends or has contributions from, you know, structures and machines inside the cell, both cell-cell adhesion and cortical tension. And we typically think about cortical tension at these interfaces between cells as tending to want to contract and make interfaces between cells shrink. And adhesion may be doing the opposite of kind of being favorable for cells to have longer contacts with each other. And then depending on the balance between cortical tension and adhesion and the kind of cell biological coupling between these machineries inside the cell, these would kind of lead to these preferred shapes of cells within the tissue. Okay, so starting with these Vertex models, around the time that we were, or just a little before the time we were starting this project, Max B and Lisa Manning were doing some really exciting work on these Vertex models where they found that there's actually a solid to fluid transition in these simple Vertex models that was characterized by one parameter, the cell shape. So if you look over here at a tissue that would be predicted to be a solid, cells prefer short contacts with their neighbors, tend to have these more rounded cell shapes. There's big energy barriers for cells to change their shape, squeeze past a neighbor and go through a cell rearrangement, so they behave like solids. And over here on the other side, cells prefer to have longer contacts with their neighbors, more elongated cell shapes. It's easier for them to squeeze past their neighbors because of these cell shapes in a rearrangement. The energy barrier drops and these behave more like fluids. And we can characterize, hold on one second, the shapes of these cells by a single parameter, the cell shape index, which I'll call lowercase p, which is just the ratio of perimeter to the square root of cell area. Okay, and Max and Lisa and their colleagues then found that there was just this critical cell shape index P0 star, which should distinguish between the solid and fluid behavior with a value around 3.81, which we'll talk about a little bit more in a couple slides. Okay, so in really beautiful work in a collaboration between Jeff Fredberg's lab and Lisa Manning's group, they showed that this very simple model actually worked remarkably well for predicting the jamming and unjamming behavior of cultured bronchial epithelial cells. And so we wondered, you know, might this also work well for our epithelial tissues inside the fly embryo, where it's really hard to do direct mechanical measurements, right? It's really appealing, right? Because you know, the idea that you can kind of look at snapshots of cell shapes and packings and infer something about mechanics is really kind of an appealing idea. And we wanted to see how far that could take us. So we went back to the fly embryos, took movies again of this germ-man epithelium during this axis elongation event. And then we segmented and found the shapes of the cells at different points in time, and then measured the average cell shape index in the tissue at these different time points. And so we have, if we plot this average shape index P as a function of time, we see that it's kind of monotonically increasing over time, and starts out kind of at a value right around 3.81 here, about 10 minutes before the tissue starts to remodel and flow. I'll remind you that kind of this 3.81 value is right around here. If we actually look at what's going on in our tissue, we don't actually get cell rearrangements until kind of quite a bit later and at higher cell shape indices, which was our first hint that maybe something was kind of not kind of quite right. And we also found, I think tellingly, and I'll come back to this later on in my talk, that if we looked at embryos of different genotypes, so say mutations in genes that are important for these different morphogenetic processes during embryonic development, they actually transitioned to having rapid rates of cell rearrangement in the germ-man at very different values of the cell shape index P. Okay, so that was telling us that this cell shape index alone was not sufficient to predict the onset of cell rearrangements in this germ-man epithelium in the fly embryo. So we kind of looked at lots of different features of packings and other factors in the tissue to see, you know, what might help explain this. I don't have time to talk about this now, but we found lots of interesting features of kind of how the exact details of the packing in the tissues, the number of cell sides and the number of cells meeting at vertices within the tissue influence the behavior of the tissue and where kind of they transition. But at the end of the day, this was not sufficient to explain our results. And I'm happy to talk about this more in the questions or offline. And so we went back to kind of a really essential feature of any converging and extending epithelial tissue, like this germ-man that we're studying, in that they're highly anisotropic. So if you look in the fly embryo, not only do we have this planar polarized pattern of myosin inducing internal stresses within the tissue that are anisotropic, we also have neighboring groups of cells in the embryo that are also kind of going through their own kind of developmental events that are pushing and pulling and tugging on our tissue. So we also have external stresses that may be anisotropic on the tissue. And so we looked in our Drosophila embryo and using a framework for analysis from Matthias Merkel, one of our collaborators, we measured how cell shapes in the tissue are aligned. And so very briefly, we triangulate the tissue and then look at the stretches for all of these different triangles and look at how those are aligned across the tissue, kind of like a nematic order parameter, but additionally modulated by how much the cells are stretched. And so we kind of look at this alignment as a measure for anisotropy in cell shapes in the tissue. And so when we did that, we found the following. So if we look at this shape alignment index, which we measure from this analysis, and we'll call capital Q, we see this transient alignment of cell shapes in the tissue right around the beginning of this process, and corresponding with this little peak we have in the cell shape index P. Okay, and so we figured, you know, we should really probably be accounting for this anisotropy in how we think about the mechanical behavior and the packings of the cells in these tissues and how those are related. And maybe for those of you that know something about Drosophila development, this time period here, where we see this peak in cell shape alignment, is occurring exactly when this neighboring group of cells in the embryo on the ventral side, the presumptive mesoderm, are bending and folding inward to invaginate at the onset of gastrulation. And it's been described by a number of other groups in some really beautiful studies that this exerts a tugging force on our germ-man epithelium over here on the lateral side of the embryo. And so we think that that's what's giving rise to this stretching of cells during this period. Okay, good. So in collaboration with Lisa Manning's group, working with a number of members of her group, Matthias, Leo, and Goncha, they decide to go in and kind of build anisotropic vertex models to try and kind of address these essential anisotropies in lots of epithelial tissues you find in vivo. And they did that in a number of different ways. One way was essentially to take the model tissue and just stretch it and to induce self-stretching and anisotropy in the tissue. And what they found was that if you look at where the tissue transitions from having a finite shear modulus, like a solid material, to having a kind of fluid-like or floppy behavior and having a zero shear modulus, the average cell shape index where that happens depends on how much anisotropy there is in the tissue. So we have a transition at a higher value of P for increasing strain or stretch on the tissue. If you kind of think about now this parameter space where we have two key parameters, the cell shape index P and this metric for anisotropy in the tissue, the alignment Q, we have different regions of the parameter space to explore. We have the isotropic case where we have low P and low Q and the tissue looks like this. We have another isotropic case where we have low Q but high P and the cells are more elongated. And then we have this region over here where we have high P and high Q. The cells are stretched and all aligned along a particular direction. And then we have a geometrically impossible region over here. Okay, so what our collaborators in the Manning group did was did lots of different simulations with model tissues with different values of P and Q and mapped out whether those model tissues behave like a fluid or a solid. And what they found was that, if we're over here where Q equals 0, the isotropic case, right, we transition from solid to fluid at some value of this shape index, which we call P naught star. And then as anisotropy and alignment increase, this transition occurs at higher and higher values of the shape index. Okay, and indeed, comparing to some previous work by Matthias Merkel and Lisa Manning with some theoretical findings, they predicted, and our simulations are consistent with a quadratic type behavior of this transition. Okay, great. So we have these predictions now for the anisotropic vertex model, and we wanted to apply this now in the fly embryo. And before I do, I'll remind you, right, we have this behavior for the cell shape index in black and this behavior for the alignment in red. And if you plot P versus Q as a function of time for an individual embryo, we start out here 10 minutes before this process at low P and low Q. These increase concomitantly in kind of a linear behavior, which we can talk about more offline. And then Q drops and we end up over here at high P, low Q. Okay, and if we look at lots of different embryos, they show the same qualitative features, but kind of shifted around due to some embryo to embryo variability. Okay, so what we want to do now is to take this PQ data and now color code each point by the instantaneous rate of cell rearrangements in the tissue at that time point for a tissue with that value of P and that value of Q. And so that's what we did here, and it actually kind of remarkably well describes our data. So plotting the shape index P, now corrected by some of these details of cell packing that I told you about earlier on, which allows us to pinpoint the value of P naught star in isotropic tissues. We see that all the data at early times sits over here on the predicted solid-like regime of this plot, and we have all blue data points indicating low values of cell rearrangement rate. And then at later times, the tissue kind of moves over here into this predicted fluid-like regime where we see rapid cell rearrangements in the tissue. And so kind of one point is that this solid fluid prediction from the anisotropic vertex model really well describes the onset of rapid cell rearrangements in the germ band tissue right at this time when the tissue starts to flow. And so this suggests that the tissue is indeed becoming more fluid-like during this process. Okay, but we wanted to kind of push on this idea a little bit more. And so we wanted to ask what happens when we alter the nature of forces acting on the tissue. In particular, we wondered what would happen if we could suppress this tugging force from the ventral furrow that we think leads to this cell shape alignment peak here. And so what we did was we looked at fly embryos that were mutants for the snail and twist genes, which are required for ventral furrow formation. And so we predicted that if we lose this tug from the ventral furrow, Q would no longer have this peak, but we'd still transition to having a germ band tissue that's rearranging and flowing. Just the transition occurs at lower values of P and Q. So we did these experiments, and if we just look here at this alignment index Q as a function of time compared to wild type in black, in these snail twist mutants, we see an abolishment of this peak in Q, consistent with our predictions. And then if we look at this same plot where we're looking at cell shape index P, cell shape alignment Q, and color coding by the cell rearrangement rate, we see that it actually remarkably well describes our data again, where the snail twist data are these kind of bold diamonds, and you can compare to the wild type data, which are these kind of transparent circles, right? So again, we predict the onset to rapid cell rearrangements in the tissue, but it happens at lower values of P and Q. So while wild type kind of moves through this parameter space in this way, moving from solid to fluid up here at high values of P and Q, the snail twist embryos that have lost this external stress that tugs on the tissue moves through the parameter space in this way. But I'll note that the snail twist embryos, these mutants, end up kind of in the same region of this solid fluid parameter space, suggesting that it's kind of going through this kind of change from solid-like to fluid-like behavior, or more fluid-like behavior in this process. So we haven't altered that. Okay, good. And so we wanted to kind of tinker a little bit further and ask what happens if we go further and actually disrupt the forces that are thought to actually drive this tissue elongation process in the germ band. Both the internal stresses from planar polarized myosin, which is essential for driving these cell rearrangements, but also some external stresses in the embryo that contribute to axis elongation as well, and driving some cell stretching that contributes to the overall process. And so we looked at another set of mutant embryos that are a mutant for Bicoi, Nanos, and Torso-like, which disrupt this myosin planar polarity and this posterior mid-gut invagination that helps stretch this tissue a little as well along the head to tail axis. And we know already that in these embryos, there's very few cell rearrangements, almost no kind of tissue elongation. And so we thought there may be a number of possibilities that are kind of at play. We could be just altering the properties of the germ band tissue, making it more solid-like so it can't rearrange and can't flow. We could be, obviously we've disrupted myosin, we could be disrupting driving forces. So the tissue may be becoming fluid-like, just like a wild type tissue, but there's no flow because there's no driving force. Or more likely, kind of given kind of the complicated nature of kind of how forces are also kind of playing a key role in determining the mechanical properties of these tissues, we thought we might be altering both the tissue properties and the driving forces. Okay. And so this is what we find. So in these Bicoi, Nanos, Torso-like mutants shown here in magenta, right, we still have this peak in alignment, suggesting the ventral furrow is still pulling on this tissue from the bottom. But Q kind of maybe relaxes a little bit more slowly than the wild type shown in black. And we can talk more about that offline. But what we see when we plot P and Q is that all of our data is kind of sitting here kind of around this predicted transition line. And all of these data are blue, where we have very low cell rearrangements. So I want to point out two things. If you look at kind of the trajectories through this parameter space over time, the tissue is becoming more fluid-like, so moving from over here to over here during kind of the germ band extension process. But we see almost no rearrangements over here. Okay, and so there's kind of two things. A, the tissue's not getting as far into this fluid regime as the wild type and the other snail twist mutants, suggesting that maybe we've altered how fluid-like the tissue becomes, potentially. And also, even though maybe we're kind of on the predicted fluid side of kind of these properties, we see almost no cell rearrangement. So suggesting that potentially we've also kind of disrupted the driving forces for this process, quite obviously. Okay, so this is consistent with this kind of third option I told you, where we're altering both the properties of the tissue and the forces driving tissue deformation. Okay, great. So I think I will kind of wrap up a bit of this story and tell you just a brief snippet about what we're beginning to work on next. So, I think all of our data point quite strongly to the fact that tissues, the germ band tissue is becoming more fluid-like right at the onset of axis elongation, which I think is a really kind of interesting general mechanism that might be at play in other tissues. And we're looking at that now, becoming more fluid-like right when you want a particular group of cells to be able to remodel and move around very rapidly during certain stages of development. If you're looking at anisotropic tissues, you need maybe two parameters to characterize the packings in those tissues and their predicted mechanics, the cell shape alignment index P, and now also this alignment index, which tells us about anisotropy in the tissue. And so this leads us to kind of, a practically useful feature of this, in that we're getting closer and closer, I think, to kind of being able to take snapshots of cell shapes and packings in tissues in lots of different contexts where it's maybe hard to do live imaging and get information about mechanics. But we do need kind of direct mechanical measurements, I think, to prove this formally. But I think the most interesting thing for me is kind of what are the molecules that are controlling cell shape, cell rearrangements, and tissue fluidity, and how are those regulated over time. And so we're doing lots of experiments using traditional kind of molecular genetics approaches and optogenetics to tune the levels and activities of key adhesive and contractile machineries. And so this, we want to know what collections of protein, concentrations, activities, localizations put us over here in the solid or over here in the fluid. And how does a tissue transition between these two during development potentially? And so we're doing lots of things. One thing I wanted to tell you about briefly is that we're using optogenetics to use light to control myosin force generation and tension in tissues. We have optogenetic tools that are both kind of controlling rho-gef localization and rho-gap localization to either predicted to increase or decrease myosin activity. And in the germ band, we can use these to control the myosin localization patterns and activity. If we illuminate just the apical surface of the tissue, the opto-gef gets us lots of myosin over the whole apical surface. Opto-gap gets us less myosin all over, and this happens on timescales of minutes. So kind of fast enough to do these experiments in vivo. We can do laser ablation experiments to prove that we're either increasing or decreasing the tensions in the tissue in kind of a spatially controlled way. And so basically what we're asking is, can we use these tools to kind of take a tissue that's flowing like a fluid and drive it over into the solid and vice versa, both to study kind of what are the proteins and genes that are controlling these kind of changes in tissue properties during development, but also as a tool for studying how, maybe morphogenesis in one tissue is related to kind of developmental events kind of at other regions of the embryo. Okay, so I'll wrap up there since I think we're short on time. We want to thank Xuan Wang, a talented grad student who did all the experimental side of the kind of anisotropy work, talented postdoc Marisol Herrera Perez, who did all the optogenetics work, our fantastic collaborators in Lisa Manning's group, without which this wouldn't have been possible, Matthias, Leo, Gonsha, and Lisa, resources from the FLY community, and our funding. And so thank you very much for your attention, and I'm looking forward to take questions.
Awesome. Thank you, Karen, so much. That was work that's very close to my heart and my interests, and it was really, really interesting. We have a bunch of good questions in the chat, but I just wanted to start with one from me, which is, clearly the external imposition of broad scale forces on the tissue are playing an important role, right, in the fluidization. Yeah. But do you have a sense of what exactly they're doing. Like, is it a strong enough force that the cytoskeleton's getting fluidized? Is it that because the cells stretch, it's sending a signal that's like, hey, you're stretched now, it's time to start doing something?
Yeah.
You know, because I'm used to thinking about these sort of individual cell-cell interactions, but how do you conceptualize the sort of tissue scale force, which is clearly important?
Yeah, so I think that's a complicated and interesting point, and I think the nature of these kind of external forces kind of pulling on the tissue from the boundaries, those aren't essential for the tissue to remodel and flow. So we can block those and we still get cell rearrangements and tissue flow. But they do seem to, there's kind of interesting hints that they play important roles in kind of reinforcing or kind of kicking off some of these events. But what seems to be really essential, at least at these early kind of 20, 30 minutes of the process, a lot of it is these kind of genetic programs which planar polarize the cytoskeleton and myosin early on. But I think it's still an open question, how the external forces are kind of being integrated by cells with these kind of internal local kind of active stresses that drive rearrangements at certain cell contacts. But a lot of it is kind of, myosin planar polarity and the genetic cues. And I think mechanical cues are playing a role, but potentially maybe reinforcing some of the molecular and genetic cues that are there. But I think that's still a super open question. I think some of these optogenetic tools will actually be able to help us directly address some of these mechanisms in a way that wasn't possible before.
And do you think that if the forces went perpendicular, that you could get them to elongate in the other direction? Or is it the program is so strong that the cells are gonna elongate?
Yeah, so that's a super interesting point. I'm trying to think if there's probably some experiments that maybe indirectly address that, but we've been thinking about doing things where we, instead of using our optogenic tools in the germ, going to other regions of the embryo and kind of tugging on the tissue in kind of different directions and seeing who wins sometimes. So
cool.
Yeah. And we can also look at that in the Vertex models and see, if those are consistent or not, or what we're missing in those models that we need to put in to bring in this kind of mechanotransduction feature of the problem. Yeah.
Cool. Thank you.
So we've got a couple of questions from Max B. The first one is, have you considered the effect of cellular rosettes in the transition between fluid and solids?
Okay, yeah, so that's a fantastic question, Max, and I know one that you've thought about, a lot. And so I'll point out, so we haven't gone into it in depth as much as I'd like to, but I'll point out, maybe I can, one thing in the simulations from Matthias and Goncha, there's some interesting things that happen where there's this little bulge in the model tissue behavior where we have, this seems to be associated with an excess number of higher order vertices in the tissue at this point. And I don't know what's going on there, but it also happens to be where the wild type tissue crosses through this parameter space. So I think there's something interesting going on there, but yeah, I think we haven't kind of thought about that as much as I'd like to. But if you have specific things, I'd love to talk more. Yeah. Let's see.
Max's second question was, have you compared the spatial organization of the active tension network between experiments and simulations?
Yeah, so that's another thing that we've been wanting to do. So actually look at kind of the myosin localization at specific edges, and it is variable between different cells, and we haven't put that in the model yet. So that would be something that would be nice to do, both looking at the myosin, but also kind of details of cell shape and kind of tensions at edges in the model and comparing to experimental data to the extent we have it. But yeah, those are all things that we'd love to do, but yeah, haven't done it yet. Thanks.
Okay, great. I'd like to invite Ashris to unmute his mic and ask his questions.
Sure. Hey, Karen. This is
Hey, how are you?
I'm good. How are you? We met a couple of times.
Yes, I know.
It's a great talk, and I do have a couple of questions. One thing is with the anisotropy in the strain in the tissue, there seems to be a new index cell shape alignment as well. And I was wondering if you can, instead of using a cell shape alignment index, can we use the strain on the tissue as the other axis? The reason I'm asking is this, is there an elasticity for the tissue as such? Because in a solid phase, you seem to have, as a strain on the in the vertex model predictions, as the strain is increased, the shear modulus or the energy required for such rearrangement increases as well. And I was wondering, is this all related to some sort of an elasticity related to the tissue deformations as a whole?
Yeah, so that's an interesting point, and one that I think maybe Matthias has thought about in detail a little bit more than me. But yeah, I'll point out that kind of if you just wanted to look at kind of the overall, were you talking about like an overall macroscopic strain or kind of a local strain?
I'm talking about like macroscopic
yeah,
strain,
so
trying to like predict the macro.
Yeah, so
you
know, I think in the solid regime where you don't have rearrangements, you can map you know, from the macroscopic tissue strain to these cell shapes in a fairly reasonable way. But once you get into the fluid, you have to be a little bit careful when you have rearrangements. You kind of have to kind of account for those properly in how you're connecting the macroscopic strain to the local cell shapes. And so, you know, Matthias, I think in his 2019 PNAS paper and in kind of in this paper kind of goes through those in a little bit more detail, how you account for that. But yeah, there's interesting features. You can look at kind of when we're right around this transition, kind of the relationship between cell shapes and cell rearrangements is kind of different than we have deeper into the fluid regime. I think getting into this idea that kind of some type of elasticity, or, you know, one way you might even think about this is we never deviate so far from this predicted solid fluid transition that maybe kind of over the time scales we're thinking it's better, you know, maybe this is kind of a weak elastic yield stress solid, and kind of the forces kind of are driving more kind of plastic type cell rearrangements if we're close to this predicted transition line. And that's not something we kind of have explicitly thought through. Yeah.
Okay. Yeah. Thank you.
Yeah. Great question.
Okay. Next up is Ankita Jha, who says, "What's the effect of cell adhesion, increase of ECAD in switching from solid to fluid?"
So that's a fantastic question, and one that I didn't get to tell you about today, but my grad student Xuan is also working on experiments where we systematically either increase or decrease ECAD levels in the germ pad and look at how that affects cell rearrangements in the tissue. And, you know, I think there's, you know, this is not something that's, I think, unique to this tissue, is that we can push ECAD levels around pretty far without really changing the rates of cell rearrangements in the tissue really dramatically. We get kind of a subtle shift where if we either increase or decrease ECAD levels, we actually get faster rearrangements in both cases compared to wild type. And so we were trying to understand what's going on, but it seems likely that what we're actually doing when we change ECAD, the dominant effect may not actually be this kind of energy from ECAD bonds in kind of this picture of line tension, but actually what we're doing is altering the organization and localization of actomyosin patterns in the tissue. And so we're kind of going after that right now, but I think that will help us kind of explain our effect. But, you know, basically, an interesting feature when we do either of those perturbations is not only do we get faster rearrangements, somewhat faster, we also get more elongated cell shapes in both cases. So it's consistent with this simple picture in the Vertex model that more elongated cell shapes are easier to deform and connected to more fluid-like tissue behavior. But, you know, the connection between, you know, these pictures of how ECAD and myosin actually feed into those preferred cell shapes, I think is still much less clear than maybe we like to think it is. And so we're digging into that now. But yeah, that's a really fascinating question, you know, what ECAD's doing here. Yeah.
Thanks, that's awesome. Do you have a couple minutes to stick around and keep answering questions, Karen?
Yeah, I'm here.
Okay, great. We some people have to leave right at the hour, but there's still plenty of people here and plenty more questions, so we'll just keep going through them. So from Tony Tsai, says, "Cool talk, Karen. What are the variables that determine the final state of the tissue on the PQ plot? More specifically, why is wild type and snail twist mutants ending up at the same final state?"
Yeah, so let me actually go to that too. So that's an interesting point. And, you know, I think a key feature in the snail twist is that we haven't actually changed the germ pad behavior. So let me see. Sorry, lots of clicks. Okay, so in the snail twist, right, we end up over here. And if we look back, maybe comparing Q in the wild type and the snail twist, Q is almost the same in both cases. And I think that's because not only do cell rearrangements in both cases help relax this kind of stretch in the tissue and in cell shapes, but also if the ventral furrow is pulling down, a little bit later, the posterior midgut induces a stretch along the other direction. So part of that may be that we've kind of ironed out some of the anisotropy, not by fully relaxing it along this direction, but actually by kind of stretching it out so it's more isotropic, again, by pulling in the other direction as well. So yeah, I think, the key feature here, I think, is that we have kind of planar polarity of myosin inside the tissue that generates the active stresses are roughly similar between the snail twist and the wild type, and the external forces, kind of everything kind of works out so that you end up in roughly the same place. And that may not be, coincidence. It may be something about the way the cells are rearranging to accommodate those changes in external stresses. Yeah, great. Does that answer your question? Or maybe I totally missed the mark.
I'm going to guess yes, but I can't speak for them.
We'll assume that was a yes.
Okay, great.
Okay, where are we up to? Pranav Meta says, great talk, Karen. Just one question. Why do the snail twist mutant cells see a decrease in their shape alignment before it goes up?
Okay, yeah, so that's, let me think. I've actually not thought about this little dip here in detail. I think we'd have to look at the data a little bit more and see if that's, strong enough to be real. It might be due to something related to other events in the embryo, in the amnioserosa, or some of the furrows that are forming that aren't the ventral furrow. I'm not sure, but that's an interesting question. And then I think some of this stretch over here is from the posterior midgut pulling. So this Q isn't telling us about direction. It's only telling us about the magnitude of alignment, but we could break that out into the X and Y contributions and look at kind of where that's coming from in more detail. That's a good point.
Great. From Ankita, she asks, would you say that the solid to fluid transition is absent during mesoderm invaginations since it's more isotropic? Okay, so I haven't, I think maybe Max has looked at this more than me in the presumptive mesoderm on the ventral side of the embryo, but there, to my knowledge, there aren't cell rearrangements occurring in those groups of cells, and even though there's kind of stretching of the cells along particular axes due to the balance between internal stresses generated by myosin and external mechanical constraints, I would guess it's kind of like later on in the process, it probably starts out low P and low Q in the solid regime, and then ends up at high P, high Q as it's folding in and the cells are stretched. So maybe still in the solid regime, but I haven't done those experiments. We're planning to kind of look at all the different epithelia in the embryo and kind of make a zoo of like where everything sits in the PQ parameter space and see if it works well or if it misses the mark once you have more complicated things going on with epithelia with more kind of extracellular matrix attachments, or kind of more kind of complicated things going on at cell adhesions. And so, yeah, we're starting to look more broadly. Yeah.
Cool. Atul asks, is the solid to fluid transition more gradual or a kind of digital response as it happens on shorter timescales?
Yeah, so I mean, I guess I haven't thought about this in detail, but it happens pretty rapidly, where you kind of over just a few minutes, you totally change what the cells are doing. And that's totally consistent with the time scale at which myosin starts out kind of uniform and then polarizes over a few minutes just before this process. So, again, I think pointing towards these cell biological mechanisms that control planar polarity in this tissue as a key driver of the changes in the mechanical properties and the forces driving it at the same time, right? You can't separate them out in this tissue, which makes some of these discussions a little bit complicated. Yeah.
Definitely. So we have a few more questions, and then just for the folks that are still here, Hawa was going to stay afterwards and answer some of the outstanding questions from her talk. So we'll just keep going until we cover everything. All right. So Karen, from Raphael Petrosian, 2 questions. First, have you looked at the correlation between cell rearrangement rate and shape alignment Q? Is there a causal connection between these?
Okay. So I'm trying to think, I'm sure we've looked at this, and I'm trying to remember exactly what that plot looks like. But the Q in the wild type germ band, a lot of the signature where P is high is from these external forces. And so, I think, and the cell rearrangements may be influenced by those, but are largely dependent on the internal forces from myosin. So I'm not sure if those correlations between cell rearrangement and Q would hold up in the wild type if you compared it to the snail twist mutants where Q is low and we still get lots of rearrangements. So I think if you look between different genotypes, the correlation might not be strong, even if in certain cases there is a correlation just due to, how events and when they're occurring in the developing embryo.
Okay, great. And second question from Raphael. From the plots you showed, it looked like the relative anterior posterior tissue length is a time derivative of the cell rearrangement rate, and the average shape index is a time derivative of shape alignment. Is this a wrong or a right impression?
I think, let's see. So I think that if you look at the cell rearrangement rate and take the integral of that, you would get a curve that kind of looks similar to the AP tissue length over time, but not identical, since there are also cell shape changes that contribute to the total tissue elongation. And then I don't know if we should think about P as a time derivative of Q or if those are independent, but I would have to think through that a little bit more carefully. Thanks.
Cool. And I think this is the last question, unless we've missed one out. It's from Wen Hu Tang. Sorry, I'm sure I'm pronouncing that wrong. Great talk. Is there a reason to look at the P with a specifically pentagon dependence? Do you see a similar result for other polygons like 6 or 7-sided?
Okay, so I think you were maybe talking about one of those earlier slides where I was looking at cell packings and how that's correlated with cell rearrangement rates. And I believe we looked at the fraction of 5-sided cells as a metric for the distribution. So, if everything were perfect, we would have all 6-sided cells here. And if, deviations from that, we spread out that distribution. So we looked at the number of 5-sided cells as a metric for how dispersed those cell slides are, even though we can fully measure. And I think we ended up using that because both in the experiments and in the simulations, the fraction of cells that were 5-sided actually gave us a really nice prediction for how this P0 star in an isotropic tissue should vary. So I think we can go back and maybe talk through that with Matthias again, but I'm pretty sure that was actually a really nice, easy to measure metric for cell packing disorder and how that would affect P0 star. But vertex coordination number also could be a very nice predictor. Max has a really nice paper on that. Max B has a nice paper on that. And we also found kind of nice correlations with the number of cells at higher vertices. The average vertex coordination number in the tissue also controls P0 star. And we don't know how those two are fully related to each other, even though they are, right, in terms of affecting the mechanical properties of the tissue.
All right, awesome. I think we got through all the questions for you, Karen. That was really, really, really interesting. We have about, 15 people in the chat saying thank you for a good talk, but we'll skip through those. So I think we're gonna, there's, maybe only a third of or less of the folks that we originally started with. So for those of you who've hung around, I'm not sure how many people are left. Is Hawa still here, actually?
Yeah, I am here.
Oh, you are here. Okay, I missed your video. Yeah, so I don't know if I've sort of lost them in the chat, but I don't know if you want to go back and
so in
ask
the
some of those
list,
questions.
I think only Mazida is left. I think everyone else is gone. Like Yolanda is still here. I don't see her or him. I don't see Atul anymore, Will or Becky. So Mazida is the only one left. I replied to her.
Okay. Yeah, I'm sometimes we've thought about trying to allot more time for this, and trying to squeeze 2 speakers in is always tricky, but we appreciate you guys working with the complicated schedule.
No, that's totally fine. I think it's, I don't know, I don't like answering during someone else talk, so, but yeah. So,
Okay,
so her
Atul's
question
here.
is, so, okay, so I will answer to Atul then after. So Mazida, your question is with regard to the citrullinated DNA being susceptible to autoimmune generation, do you think the PAD4 activity is required to enable the release of the net or serve to attenuate the damaged response? So what we found is that the activity of PAD4 is required for net release. So, and for the attenuation of the DNA, of the damaged response, I would actually think that it would worsen the damage response, right? Because if it citrullinates a protein, and those citrullinated protein generate autoantibody, so you will have something that the body can generate an inflammation from. So,
hi, Hawa.
Hi.
Yeah, but that's exactly what I was thinking. That if it's bad, then why is it really required? Because in apoptosis, you get condensation to protect the DNA so that you don't have single strand exposed or even double stranded DNA exposed. So is it something to do with the release that it needs to be decondensed in that specific case, or?
Yes, so the decondensation step is required for the release of the DNA, both outside of the nucleus and outside of the cell.
And the citrullation helps with the decondensation.
Exactly, yeah.
So you can think of a case for blocking the citrullation, but actually still having like histone deacetases or something to
exactly. So if you so if you treat cells, if you pre-treat cells with an HDAC inhibitor, so this drives DNA decondensation, and you can induce the rupture of those membranes. So decondensation is required, and I mean, in my opinion, like, I don't know how common that will be, but no matter what protein is doing it, once you reach the step where you decondense the chromatin enough, you can generate enough force to rupture those membranes. And Claire will kill me, but I think the proteins are details. So, but that's my opinion.
So you'd say for the cases where these cause more autoimmunity, actually blocking the PAD4 and like maybe treating with HDACs inhibitor is actually a
potential way
forward.
Yeah, that's what I would say.
Yeah. Thanks so much.
Thank you, Mazida.
Bye.
Bye. So Atul, your question is whether actin disassembly correlated to DNA release into cytosol with respect to time. Actually, no, actin disassembly is much earlier. It happens usually 2 minutes before the vesiculation, and the release of the DNA in the cytosol happens usually 23 minutes after. So there is a sort of a 25 minutes delay between the two events. I hope that answers your question. And I think that's it. There's just one more question from Will. Becky, I don't know if you... So Becky is still here, right?
She might have had to step away for a minute. I think she's still in the lab.
Okay, okay.
So, yeah. Okay, yeah. Do you want to call it?
Yeah, I will.
Okay. All right, awesome. Thanks to the few people who are left, and this is all also on the YouTube, so folks will get to hear your answers forever. Okay, so thank you.
Oh God, my proteins are details is in YouTube. Oh God,
I'm gonna use that against you forever. Yeah.
No, I love it. I think you probably have actually a sort of a good audience for that particular statement here right now. All right, I'll end the YouTube and